We're back. Everyone's
Someone's going to have to figure it out, that's for sure.
How you doing, man? I'm good. Hey, do you want a chai latte?
Howdy, folks! It is my distinct pleasure to introduce Dr. Jeff Grimm. Over the past 2 decades, Dr. Grimm has studied how craving and drug-seeking behaviors are learned, how that learning persists and intensifies following long periods of abstinence, and how it can be reduced by experiencing an enriched and novel environment. His work can be used to understand how neurological models of craving and addiction apply to a wide variety of reward-dependent behaviors. For example, while Jeff started his research with cocaine, he has since experimented with antipsychotics, nicotine, and speedballs. Is that
yes.
Cocaine and heroin?
Not personally.
Okay. Those are pretty
That's
rad.
what we call cocaine and heroin.
Oh, okay. Most recently, he's been investigating the addictive nature of sugar and non-nutritive sweeteners. When you can't put a price tag on good research, Dr. Grimm has received nearly $2 million in federal funds over the past 15 years for his work. And personally, what I appreciate the most about that is that he uses this high-quality research program as a chance to provide research mentorship opportunities for Western students. He involves student researchers in all aspects of his work, including conception, including running behavioral and biological assays in terms of analyzing data and writing up results. Over, I think, the past decade, Dr. Grimm has included 55 student co-authors across 15 articles in top-tier peer-reviewed publications. So the fact that he gets to bring this research to all y'all is part of what makes him so special. So without further ado, it's my pleasure to introduce Dr. Jeff Grimm, who will be discussing preclinical trials on craving at WW.
Okay, thanks for coming in on this Thursday afternoon. Who knew what was going to happen this morning with the snow? I live up at a couple hundred feet, so I was like, oh no. Here we go. But it's looking nice and spring like out there. Thanks, Dr. Grimm, for introducing me, and I see some of those students here that are currently working in the lab. And when he was saying, like, I provide opportunities and like this stuff, I need those students working in the lab in order to, you know, be able to collect data and be able to write papers that involve students and all that kind of stuff. So it's really a collaborative effort, like he was saying, and so cool. Come on, you guys. Cool. So this talk is, well, you'll be seeing a couple more of these before the end of the year. Dr. Zoff over here, for example. So the idea is that for faculty from the Department of Psychology who have gone on a sabbatical, otherwise known as a professional leave, the next year at some point, we're trying to encourage them to give a presentation of things that they did or happened to them during that time when they were away. So if anybody happens to have noticed, last year, winter and spring, I was not actually teaching classes. I was around though, so I don't have the most exciting presentation. Some of you might have gone to Dr. Trimble's presentation a while back, and he was in Scotland for that entire year, and he had amazing photographs of, you know, the architecture and all that kind of thing. You're not going to see that with my talk. You can just Google Bellingham and see all this. So with that idea of having a presentation about professional leave and what occurred, I do have a little part of this that I was aiming a little bit more for maybe junior faculty, and also students. Yes, I'm going to kind of like, to give you a little bit of education on this whole thing about what professional leave is. So you're trying to schedule a course and you realize, so-and-so is not there, it's not being offered a particular course. Where are they? What are they doing? Well, we go through this process of applying for leave, and we have to basically work up credits in a sense. For every couple of years here, we get a credit that can go towards this process. And then the state of Washington provides opportunities. We can apply for leave using those built-up credits. And just to give you an idea, this is not data from our lab or anything. These are going across time. The blue here indicates the need, the requests from faculty to be able to take a professional leave and do their scholarship outside having to teach, for example. And here's what's actually provided by the state. So it's a pretty good, the state is there, it's offering those opportunities. But you can see there's definitely some years where a lot of people apply and they're probably not going to be able to get what they're asking for. So it's something that as faculty we always have to be kind of hoping. There's a little bit of gamble and when you're making these applications, and a lot of hope and sometimes some despair for people. But this is the third time I've done it. Every time it's worked out for what I've asked for, I just kind of hit, I think, some of the dips going across time. And so, going back a couple of years, I was looking ahead, and I knew I wanted to take my leave. I had 2 quarters available. Winter 2018 and spring 2018, so I have to work backwards here. So in order to do that, I'm not really going to be impressing the undergrads too much. They'll be like, whatever, you got to take a leave. But there's a lot of looking ahead for this process. And maybe that's a good thing for yourself when you're looking at projects, things to do at the end of the quarter, or things you're looking ahead for graduation. A couple years later, you got to be always thinking, what do I need to do to get to that point? So you have to keep in mind there's an application due date. That was in October of 2016. This is when the leave would actually be. This is when the application was. So really looking ahead. You have to be thinking way in advance, of course, when the application's due. It's not something you do the night before. It takes months as you're considering what you're actually going to try to do. You might have to make some contact with other colleagues around the country or around the world, depending on the kind of projects you want to get involved in. So I was looking at maybe back in winter and spring of 2016. Were you guys born then? And once that started, the process of actively putting the application together, getting that to the departmental chair, and then it works its way all the way up through the provost's office, and then finally getting the approval winter of 2017, and waiting. I didn't have a fall, I didn't apply for that, and then actually doing the leave winter 2018 and spring 2018. So it's a process. I'm not complaining, I'm not whining, but just reiterating that it is a process. You have to be always looking ahead. So sometimes you might go into your professor's office and they're irritated. That's usually me most of the time. Sorry. But they might actually be working on their professional leave application for 2 years from now, and they're right in the middle of some really intense thought or negotiation online or something like that. So what we really need to be careful about, and this is again for you guys when you're looking at applying for different kinds of opportunities, whether they're scholarships or looking ahead at graduate school, is always read the instructions very carefully. And so for those of you faculty that haven't done this before, if you're looking ahead, it's really laid out. It's very clear what you're supposed to do, and it's really to explain what it is you're hoping to do and how you're going to do it. And so for me, what did I want to do? Well, I really do like teaching. You guys are great students, and hanging out with you and all that stuff, but there are a lot of things that need to be done in the background, so a lot of the writing and the working with data. And so to have a little bit of a break from being in the classroom was something that I was looking at as a way to be able to process a lot of the backed up data from our lab and start to actively look at things that could come in the future, projects that could become available in the future. And so as Dr. Grimm was saying, we've had grants in the past. I have a grant that's actually ending the end of this month, end of March. And luckily, getting a little bit ahead of the slide here, a new one is starting on April 1st. I think it's not a joke, but that's what I said. It's going to start. So go back again to 2016, looking ahead. I knew that this grant was going to end the end of March 2019. I knew I needed to renew that grant. This is through the National Institutes of Health. To do that required a lot of, front-end kind of stuff, like figure out what you want to do, who might it involve, there's going to be collaborators or mentors. And so that's when I was thinking the professional leave was how to fit a lot of those activities into that time that would be where I could have some relief from being in the classroom. So that was really kind of the core component is this NIH grant. Looking at the 2 quarters of possible leave, those were my credits I had available. I knew I could fit that in, but also some other cool things that are harder to do when teaching, like visiting a colleague at another institution, giving a seminar, talking about data, that kind of thing. So I wanted to fit that in somewhere, and I've been talking with a colleague at University of Wyoming for several years about doing something like that. So okay, I'll kind of fit that into the puzzle. And also wanting to write a review article on some of the enrichment kinds of things we've been doing in the lab. So those were kind of the ingredients, and I had to find a way to fit that, to map it on to those 2 possible quarters. So this is what I came up with for my sabbatical, the professional leave, was to have a quarter where the first bullet on here is what we're always doing. The lab's never going to stop. That's like number 1. Even if I had gone to Scotland or whatever with Dr. Trimble, the lab would still have gone on. So that's always going to be there, the mentoring and making sure that all the things that are needed to be ordered and all that stuff is done. So for the first quarter, though, is to finish this NIH grant because there was a particular deadline in February of that year, prepare for the seminar, and then start working on this review article. So that was the idea. Carrying it through, very important for designing an application of any kind like this, has to make sense. Why are you asking for 2 quarters? Well, this quarter depends on the first year. What happens in the first needs to carry over. So that was a big part of asking for 2 quarters. Over here, continuing, going to perform the seminar at the University of Wyoming, talk about research, all that good stuff, and maybe submit this review article. What actually happened, though, is another story. Nothing bad happened, but life intervenes, things happen. You have to, you're planning 2 years ahead, and then this actually, you get this leave, and then the opportunities, this, that, and other thing. Good things happen. Sometimes you have to make some choices. So here's the same quarter 1 and quarter 2. You still got some check marks. So those are the things that I did. For example, in quarter 1, got the grant in there, and eventually it was awarded. So hopefully April 1st, things will be good. I did not work as far as I would like on the review article because it turned out we had this other big data set that was morphing into a manuscript, and in kind of my hierarchy of needs or whatever, psychology joke, that jumped to the top to get that in the queue to get published. So a lot of extra work went into that. And you can see there's a check mark over there, so that actually, that article ended up getting published. So it was kind of a nice switch out. So the review article has been on hold, but now I'm working on that many months afterward. To explain these pictures here, if you haven't been to Laramie, Wyoming, and you've been way out there, you've been like that? So they're really into like horses and stuff. That's the Lucky Bronco. I was there in March, and when I flew into Denver, it was 70 degrees, and then I ended up having to drive to the flight screw-up thing up to Laramie. It was really warm, balmy, and by the morning I left, it was snowing, and I drove back to Denver in a blizzard in this tiny, one of those little tiny rental cars. I almost died. I Bluetoothed the phone to the rental car, and I called my older daughter, and I was like, just somebody needs to know. They don't show up. I didn't take that picture, but that's just from Google, but that's what it looked like. I also put up here, this is a famous bar. So did you ever go to the Buckhorn bar? Yeah. You know what I'm talking about?
Yeah.
It's got a bullet hole. It's got the whole, like, cowboys and guns and horses. So, anyway, so that was basically my professional leave, is those 2 quarters. Hodgepodge of interesting things going on. Some results here. Doesn't always happen, so I was really glad to actually have that publication come through. Get to go to UYO and meet some really cool people. go to the Buckhorn Bar. It was like 4 in the afternoon. It wasn't that exciting. But crazy stuff happens there. So that's my professional leave portion at the top. Thank you very much. Now you guys know, at least when faculty talk about sabbaticals or professional leave, it's this thing that you apply for, it's competitive, and it's time away where you're not actively in the classroom, but you're doing other scholarships. That's your take-home quiz. So this grant thing, that's the researchy part that was submitted in that first quarter of leave, and then that's a process in terms of the time frame that's involved. So when actually the announcement that was funded, that was just in January. So that's just a couple months ago. That's the time frame from February of 2018 to January of 19. And that was the best case scenario for the time frame for an NIH grant. So some people might say that's really short, but it feels really long when you're waiting to find out about the financials, that kind of thing. So here's the title of the grant: Sex Differences in Reinforcement, Role of Environmental Enrichment in the Orbital Frontal Cortex. So what I want to do next is try to take you all from the little bits and pieces of information and rationale to this particular title for this grant. Like, why does this make sense? When a reviewer sees this, or when they saw it, they go, oh, that's interesting, that makes sense. Why somebody would propose to look at these particular things. And so I'm going to start with some background, of course, to pull you in, to help you know a little bit more about things that we've been doing in our lab for the past many, many years. It's been many, many years here at Western, not in a bad way, I didn't mean it to sound like that. I came here in 2001. Right before that, I was in Baltimore, Maryland at the National Institute on Drug Abuse for 2 years. And then I was at WSU Pullman prior to that for graduate school in experimental psychology. But it was at WSU that I started working with cocaine.
In a professional capacity.
In a professional way. And rats, and operating procedures, so rats pressing lever, getting cocaine intravenously. And looking a lot at learning, so conditioning. And so when I'll show you a couple pictures of food in a moment here, but the idea is the rat makes a response and they're reinforced, right? Yay, Skinner. For the cocaine, they make a response that it gets delivered intravenously, like I was saying. But you can have stimuli presented at the same time, like a light and a tone. So there's classical conditioning happening as well. So associative learning, yay, Pavlov. Okay, so we're looking at both of those things. For an addict, and you can argue like, drugs, but also maybe food-related kinds of things, we become very conditioned to things that predict the availability of the reinforcer. So packaging, advertising, smells, places. That's how we, the Starbucks coffee cups. What do they got one today? These are just the plain when people are going to withdrawal. They don't have the conditioned stimuli present. But those kinds of things we pair with the availability of these reinforcers. So all through graduate school, looking at cocaine, went to NIDA, National Institute on Drug Abuse, same kind of stuff. A lot of cocaine work, speedball, one study with that, heroin just by itself, and then a little bit of sugar work there. And then came to Western, and a little bit of cocaine work here initially, but since then, sugar, all about sugar. So the reinforcer is really important in various ways, but it's also across reinforcers. You see some of the same behavioral effects we'll talk about just in a moment. So a little bit of background with that. Some pilot data, again, relevant to this grant application. And then I'll list the specific aims of the grant. So when you're looking at grants, you always have some kind of, this is what I'm planning to do. That's the specific aims. NIH uses that terminology, and I'm assuming a lot of the other agencies do it as well. So animal models of relapse. So rats with cocaine, rats responding for sugar. Who cares about that kind of stuff? Well, we're interested in our lab and in lots of other labs and our funding agency, like NIH, in human problems. So people take drugs recreationally, and sometimes that's not a big deal, but sometimes it can become quite problematic. So people become addicted. I'm doing my quotes because there's a lot of definitions I have to get into for that. And one feature of that addiction is relapse. So you try to quit, and maybe days, months, years later, something happens and you go back and actively start pursuing drugs again. Or maybe you could argue food. So you try to go on some kind of a diet, and you stay away from things for a certain amount of time, but then you relapse, or sometimes people refer to that as diet recidivism. Same idea though, certain cues in the environment can actually get you to go back when you're not really intending to. And it has many negative consequences associated with it. So that's our big translational idea, is that hopefully whatever we find in our research can help inform about what's happening with the behavior and in the brains of people, including ourselves. There's always often a looking in the mirror kind of thing, especially with sugar, I think, at least for myself. So we're very much interested in craving that's induced by cues. Craving is another one of those air quotes, like, what is that? Controversial in literature in terms of operationally defining that. I put a couple words there, like urge, desire. For humans, probably a lot of that's happening at the subconscious level. I could easily go on a tangent there. But at some point trickles up into the consciousness, and then you feel like, oh, I really want those cookies. Somebody ate a lot of little sugary ones, and like, hopefully they'll be left over. But exploring all this with the animal model, we use the rat model. Other laboratories, a lot of laboratories use rats. Some use mice. Very, very few, I always like to mention this to students, very, very few use non-human primates. They are still used for research. Chimpanzees are not used in animal research like this, but they're usually smaller monkeys like macaques, just so you know, are aware of it. It's a very small percent of the research that's done. But again, we work with a rodent model where we can try to examine what we're doing now is food intake and craving, but as I said previously, work with drugs like cocaine. Here's one of our friendly little subjects in an operant box. And so this is a place where we can control all of the environmental features for that animal. I know some of you in classes have explored this a little bit more maybe than others. And so you can see there's a lever over here that they can press, and that's going to activate a pump that's hidden off the screen that delivers, for this rat, it would be sugar water, come out of this little bin over here. You can also see the Pavlovian stuff happening here. Here's a light. You can't hear it, but there would be a tone. So we can have different stimuli from different modalities paired with the availability of the sugar here, or if it's a drug study, again, they have a different setup to allow the intravenous delivery of the drug. So cues we can activate. Other stimuli that might be something like an occasion setter, a discriminative stimulus, other kinds of things you can look at. For behavioral studies, we also have this funny lever over here that doesn't do anything, but rats like to do stuff. They're always kind of busy. So that for some of our studies, we make use of that a lot if we administer a drug to see if they've just gotten really hyperactive or something like that because of the drug, or if they're heavily sedated because of the drug. We can see if their kind of general activity changes. And we also have, it's not shown on here, but we have photo beams to detect their overall just movement, so their locomotor activity. This is a very quick, I'll just show a little part of it, just to give you more of a visual. That's the rat being placed into an opera chamber. I've got the volume turned down, you don't need to listen to that now. It's a really big rat. Some of ours are typically much smaller. So he pushed the lever, he got some sugar water, oh, that interactive lever doesn't do anything, that's boring. And then we collect the data, so we have a computer system, collecting the data, runs the program, sets up the requirements for what the rat needs to do in order to be reinforced, all that good stuff. This was a video actually made as part of many years ago now, of a project that's published online if you want to see the whole thing. It's got students from Western in it, like reading the cue card. This research is really they were so good. Okay, so that's the basic kind of procedural stuff. Rats in boxes, pressing levers, getting reinforced, and some classical conditioning, operant conditioning, all that good stuff going on in the background. So things we've looked at before that are important for craving, and Dr. Graham mentioned one of them is how craving changes over abstinence. We'll talk about that as the incubation of craving. And related to that, we did some manipulations. I'll show you some data about the resistance of incubation to some of those manipulations, including satiation, and also this environmental enrichment stuff, which we're doing a lot with these days. And talk about a little bit of the neurobiological findings from the lab over the years. This is really just kind of selecting a few kind of highlights that were, again, part of the grant application, so more specific to that, building up to that rationale for those specific aims. Okay, so here's some data. This is made up. This is just a schematic. This is what behavior typically looks like for rats that are being trained. So basically, we say they're being trained, but we're just giving them the opportunity to respond on the lever for something. So you can imagine they're responding for cocaine daily across these training days. Or maybe it's for sugar across these training days. You can switch out the different reinforcers. Basic idea is the first couple of days they're learning, oh, if I do that, I get this good stuff. Oh, if I do that. And after a few days, they get really consistent at that behavior. One of our typical dependent measures is just how many times they press the lever. There are other things we look at as well. But this is like our kind of prototypical dependent measure for looking at how much they're willing to respond for the reinforcer. And then also when we take the drug or the sugar away, how much they're willing to respond in its absence. So more that craving, like, oh, where did it go? That kind of thing. So across training, they have the reinforcer available when they press the lever. And then for testing, over here, about day 1 and day 30, they no longer have the reinforcer available. They're pressing and they're either getting nothing, depending on the way we do the study, or they're getting those Pavlovian stimuli, the light and the tone. But they're not getting the reinforcer anymore. They're not getting the drug or the sugar for the right-hand part of the graph. So that's testing their craving, their seeking behavior. And so with incubation, the general phenomenon is that the amount of responding that occurs very early, since it's been their last experience with the reinforcer, the amount that they respond very early versus very late in abstinence, we usually call it abstinence, they're away from the reinforcer. The longer it's been, the more they'll respond when they come back to that box. So they haven't been in the operant box, they've been back in their home page, but they haven't had the sugar, they haven't had the cocaine. This is their kind of our prototypical is having it at 1 month, and that's what we saw initially in some of our first work with cocaine. So the behavior actually increases with abstinence. This was the first paper, this was from when I was a postdoc, so this is at National Institute on Drug Abuse. This was with cocaine, where we came up with the term incubation of craving. We brainstormed. What are we going to call this? Because it seemed to be, for some people, opposite to what they expected. And, so there was some head scratching and like, what? I thought if we just waited long enough, the abstinence symptoms would decrease, they'd diminish. And our rats were saying the opposite. And so we were confronted with, well, this is an interesting behavioral finding in an animal model. It's counterintuitive. Let's get it published. So without getting into the detail, you can see it goes up over the course of abstinence. There are a couple different ways of measuring it. They're not responding for the actual drug. They're just responding for the cues that were previously associated with the drug. You can see it's this nice linear increase over time. There's been a lot of follow-up studies. So including with sugar. This is our first study we did here at Western with sugar. You can see I've got the arrows here to totally guide your visual tracking. But with the white bars here, these are the basic measures of craving that we use without getting into all the details. Again, you can see over a course of a month, responding increases. So same with cocaine, same with sugar. Other laboratories have looked at this, including the lab where I was a postdoc. They've published a lot on it since then. But other laboratories around the world have done some work with this. And so you can see the list has grown from cocaine, sucrose to alcohol, heroin, methamphetamine, nicotine. Saccharin we did here. Some other food stuff we've done in other labs. And even just plain water when they're not even made to be thirsty. Just when it's available. over a period of not having it available to an operant response, and actually their motivation to respond for it increases. So we're like, what's up with this? Is this just a weird rat thing? Well, it turns out over these years, since 2001, when our cocaine paper was published, other laboratories who work with people have said, hey, we're either looking at our data that we already have, or we're actually we just conducted a study where we looked for this explicitly, and they have also seen incubation, but again, with people, with human people, right? So for cocaine was one of the first ones, but also for alcohol, heroin, methamphetamine, nicotine, those with smokers, and with food. So we think we're onto something again, the goal of translation, of trying to understand a human condition, having this model system, the animal model, so we can look at that, we can work with it, do different kinds of manipulations that might better inform us about what's actually happening with people. And so this incubation, if you think about it, if you're a person and you're trying to remain abstinent, what these data actually indicate is that the longer it is, at least to some point, we don't know where where things might go back down for people so much. We know for rats that it does tail back down after several months. But imagine you go into treatment, you go in for a 2 or 3 day stay at some kind of inpatient facility, and you detoxify, you get all the heroin out of your system or the alcohol, or maybe you try to work it with sugar or something like that and refrain from all the sugar. A lot of people try that in Bellingham kind of a thing. And then a few more days go by, or a couple weeks go by, months go by. It could actually be that the longer it's been, the harder it's going to be to remain abstinent. Those cues in the environment that are calling out to you, they're reminding you of the drug or the food, could actually become more potent over time. So it's a troubling finding, actually. Sorry. But it's good for people to know. Knowledge is power. All that kind of good stuff. So back to the rats, a couple of the manipulations, things that we found for background here about the satiety manipulation. I won't go in crazy into detail, but just the same graph we saw a minute ago. I was having you focus on the white bars. Now instead, look at the gray and the black. For those conditions, those are different rats, went through all the same training, the same amount of abstinence, but for the afternoon and the night, all the way up to when they were tested for QE activity, they had a big bottle of sugar water in their cage. So they could just guzzle down. It was like having 2-liter bottles of Coke or something like that. Coca-Cola. Drink, all the sugar they want, and they did. It's a different data slide. So lots of consumption of sugar overnight. Then they're confronted with the ability to respond for sugar-paired cues. You would think that maybe after all that sugar, they'd see a sugar-paired cue and be like, I'm not interested, I'm not going to press that. We actually saw a little bit of that here, if it's only been 1 day since the last time they had their training. But if it's been a month, even though they had sugar the entire afternoon and night before, it had no significant effect on their behavior. If anything, it almost promoted seeking. We even did another manipulation that's not on the graph, where they had sugar for 3 days, just a bottle of sugar water, all they wanted, guzzle, guzzle, guzzle. Then they go back in and saw the cues, and they still responded for sugar-paired cues. So the incubation was still there. So this is like a negative finding for the most part here with satiation. So we couldn't break incubation by letting them have all the sugar they wanted. So we've tried different kinds of manipulations to try to reduce incubation. A lot of them have been behavioral pharmacological, so administering different compounds like either agonists or antagonists of particular receptors. And we've had some luck with a couple of those. Those data didn't really fit into the rationale for this particular grant, so I'm kind of skipping over those things right now. But one thing we have found that is very effective, it's very robust, the finding at reducing sugar seeking and including incubation of sugar seeking or sugar craving, is to allow rats to experience environmental enrichment. So that's another vague thing, like what on earth is that? And in the literature, it's actually like, you have to be really careful when people talk about what they're doing for environmental enrichment, what does it mean in one lab versus the other. It's still in that kind of state of not very cohesive in terms of the operationally defining what people do for it. But overall, the effect across labs has been pretty consistent when it comes to reducing craving. So this is what we do. Again, a little bit ahead of things here. Rats trained up, responding for sugar over those. We typically have 10 days of training. And then they were allowed to live in this enriched environment. In the first study we did, they lived in there for about a month. And then we looked at their reactivity to sugar cubes. So this is what we use. This big old ferret cage from a company in Portland. This is their normal home page, by the way, just for comparison. So it's this big cage. If they're in the control condition, it's one rat, so they're somewhat isolated. This is the typical condition. EE, they've got friends. There's usually either 4 or more recent studies, we have 3. This isn't tricked out as much as we usually have, but there's usually a lot more toys in there. There's multiple water bottles, and the toys get refreshed 3 days a week. So there's a lot more handling that can happen with that, but also the social interaction, the novelty, all the stuff that's going on, all the stuff they can do in the EE environment. So what we found in the first study, this was many years ago now, published in 2008, but I think we started on it around 2006. It actually started from one of these, you guys over in the building over there might have seen these flyers for undergraduate research projects through RSB. It started through one of those. I got like $500. I went to the toys, not the toy store, but the pet store to buy bought all their toys. And that's where that started. For this particular study, what we found, I'll just focus on the main big effects here. If you look at this is one particular measure for the rats after they've been in there for a month. Now they're confronted with the ability to respond for sugar cues. The ones that have been enriched, much less. For a different measure, what's very specific to the tone and light cue. Again, very big difference between the controls and the enriched. So that's 1 month of enrichment. We decided it took a little while actually to kind of gear up and follow up on that. But we ended up doing a big parametric evaluation where we looked at, is it just having friends? Is it just having a big place? Is it just the novelty of it all? And that's what all these different groups are here. And we also included incubation as part of this. So to be able to look at their behavior on day 1 of abstinence, that means they can only be enriched overnight from their last day of training. So that's why we started exploring this acute enrichment thing, just being in there overnight, versus having a condition where you look at their behavior 30 days into abstinence, where the rats have been in enrichment the entire time. So that's this group right here, which is similar to the previous slides. The rats have been enriched for a month. So we replicated that main finding. So here's controls, they've been enriched for a month. But you can see there's other conditions where different aspects of enrichment were also successful at reducing sugar seeking after a month. One of which, the most robust, is just being in enrichment overnight. So we call this acute enrichment. You can see there's one up here as well. And so that's just about 17 hours of enrichment. So it's late in the afternoon on one day, and then testing is into the morning of the next day. So it's the same apparatus, but the timing is different. So here's the acute, if it's only been 1 day since the last time they were trained with sugar, and here's where it's been a month. But just the night before, they were in enrichment. So we know that this is very effective, as is chronic. If anything, the acute enrichment is a little bit more effective. Like, okay, what is up with that? We need to explore that a little bit more. And so again, some of the behavioral pharmacology, different kinds of things we've looked at. One question has been like, what's happening in the brain? Like, where in the brain is this being mediated? So we've had different kinds of questions about anatomical specificity with that. And with that design of having like a day 1 and a day 30, acute enrichment, or chronic enrichment, you basically have these 5 different conditions to look at. Enrichment with or without abstinence, that kind of a thing. So with that in mind, starting to explore brain substrates. So if you haven't seen schematics like this before, this is from an atlas of the rat brain. And there are different ones for mouse brain and other critters, and we've explored many different brain areas. I'm just going to focus in on the orbital frontal cortex because that's the area that ended up being part of the specific aims for the grant. In a rat brain on a slice frontal section like this, the orbital frontal is basically this area here. For us, it's just behind, like up above the eyeball on each side. That's how the orientation is for the human brain. For rats, it's a little bit different. But this is the area, orbital frontal cortex, who cares? Well, it's really important. It appears to be involved in a lot of things. I'll be totally honest about that. The literature is pretty messy and around the edges. But definitely where things come together across all sorts of kinds of reinforcers, the orbital frontal cortex helps us to assess how important a particular reinforcer is like right now relative to others that might be available. So kind of weighing like, should I focus on this or should I focus on that? And it helps to guide you towards the one that's the most valuable at the moment. So if you were to do a search on PubMed or Google Scholar or something like that, with brain imaging in humans, orbital frontal cortex comes up a lot with addiction things, including things like gambling, teenage behavior, all these kinds of things. It shows up, the orbital frontal cortex. So this first study, published in 2016, we used a technique called immunohistochemistry. So that's where we're looking at slices of tissue and looking for this protein FOS that is expressed in neurons that have just been recently like excited, like something turned them on, literally. And so this protein piles up inside the cells. You can use an antibody to detect in the slice. And so these are some examples here, these little brown colored deposits in these cells. So that's the FOS, how it shows up. You then count those cells and then compare across your treatment conditions. This study we looked at, was it 18 different brain areas? Oh my gosh, that study. I was seeing spots for like 3 months in the microscope after that. So lots of data, we found a lot of interesting things in different brain areas, but focusing on orbital frontal cortex, very salient here is the fact that enrichment, not only does it lead to less craving behavior, less sugar seeking, but way less, it sounds very scientific, FOS immunoreactivity. So a lot fewer cells that are saying, hey, this was an important thing going on at that time when the brain was collected, whether it's the acute or the chronic enrichment. So FOS is saying something's happening with enrichment. We also, this was the study I ended up spending time getting published on the professional lead. We also looked at this protein called DARK32, which gets a little bit more complicated getting into the details of it. You can see this schematic. It's like, oh no, there's like arrows and stuff. The protein is here in green, and basically it's a protein that can be affected by stimulation at different receptor sites nearby, through like dopamine receptors, for example. That's kind of the primary means of messing with it. That's why it's called dopamine and cyclic AMP regulated neuronal phosphoprotein molecular mass 32 kilodalton, DARK32. But dopamine definitely important. But also the neurotransmitter glutamate can interact with its particular activity state. An activity state for proteins usually it means something's happening with a phosphate group that's been added or removed from it. So this protein can be phosphorylated or dephosphorylated at several sites. There's one site at a threonine amino acid is the one that seems very important and ultimately leading to changes in levels of FOS. So it's a part of the neural process, the intracellular process of the cell responding to what's happening at its receptors on its surface that's saying you should be really excited or not. So eventually you might end up with some FOS down here. So we're interested in the phosphorylation state of DARK32. Same kind of a study. By the way, this is the protein. This is where you go, oh my gosh, I gotta figure all this out. So if you want to look at this in more detail, that's what Google's for. Now, I could always talk you through it later on. But these are different sites for phosphorylation. And so, for example, through the dopamine D1 receptor, we've done some behavioral pharmacology with that. You can see the arrows eventually lead over to FOS. So it's in a place where it can kind of help modify signaling to push the cell to be more excited or less, including as part of being more excited, maybe change, maybe adapt, part of neuroplasticity to be differently responsive to stimuli in the future. So again, lots of data. We did look at 18 areas. We looked at 9 this time, and ended up finding interesting things in 8 places. That was cool. But here it's just focusing on orbital frontal cortex. You guys now know about incubation. So we saw some changes in DART32 phosphorylation related to incubation. So that was interesting. So we didn't see that with FOSS. But we also saw some changes related to environmental enrichment, including kind of an increase if it was the acute enrichment for day 1 of abstinence, but also for the day 30 when there was acute or chronic, we saw a decrease in the activity of this protein that, again, mediates signaling on the cell surface, the neural surface, mediates that signaling, telling that cell how to maybe adapt to things in the future. So like, okay, orbital frontal cortex seems to be saying to us that it's important in these EE effects. We don't exactly know why or how, but let's maybe explore this a little bit more. So brief summary, incubation we talked about, EE reduces that, whether it's acute or chronic. And there's things going on in the brain, vague there, including in the orbital frontal cortex. So let's explore that with this grant proposal. Yay! And then you're like, sex differences. Where did that come from? Well, let me briefly hit that last few minutes here. So this is the proposal. Are there sex differences in the behaviors related to consumption of sugar? So working to get it, as the rats do. And are there sex differences in the effects of environmental enrichment on sugar taking, sugar seeking? And then can we mess with the orbital frontal cortex to learn more about how environmental enrichment decreases those sugar-seeking and taking behaviors? So the sex differences thing, a lot of blah, blah, blah here, but I'll hit the main points. There's a lot of history in clinical research, for example, of research being done primarily with males. This goes back decades and decades. So, you know, how we know how the heart works and all this kind of stuff, primarily because of research being done for the clinical side of it in males and also in the preclinical or the animal model stuff. So a lot of history there. For clinical work, though, over the years it's gotten better, and some people might be surprised that currently, this is through NIH, it's about 50-50 now for representation, males and females, so that's been, you know, kind of more, makes a lot more sense what we're looking at. The preclinical work, the animal model stuff, has not caught up yet. I don't have a number for that, but I can say that primarily work done in rats and mice, and even monkeys, the little amount that's actually done, is still primarily done with males. And so there's a lot of problems with that, but the ethical kinds of things, like we're gathering all this knowledge, but it can't be necessarily shared with at least 50% or more of the population if you're trying to translate the importance of the work. So the generalizability is not great, et cetera, et cetera, et cetera. Plus, so there's a carrot and a stick kind of thing. We want to learn more, but also NIH is like, if you want to get funded, you have to start doing this. And so there was a policy just a couple of years ago called the Policy on Sex as a Biological Variable. You can see here is a mandate. Like, if you want to get funded, you have to either do this or explain why you're not doing it. So if you're doing research on ovarian cancer or something, then, well, we probably wouldn't include males or something. So there are ways out. But beyond that, it's been a big shift in funding and grant applications and people kind of scrambling. But it's a really important thing for our work because in the addiction field, there are definitely some major sex differences in both drug addiction and food-related kinds of behaviors. So craving behaviors tend to be more intense in females, the leading to relapse, food, they have up here eating disorders disproportionately affect women. I put lots of dots there because there's a lot more. I could be able to lecture with that. So keeping that in mind, we've been doing work on sex differences as part of leading up to the grant application. And we started, let's just look really simply. What do the males do when they have access to sugar compared to the females? So we've looked at behaviors on what's called a fixed ratio and a progressive ratio, schedule reinforcement. The FR is just the typical, you make a response, you get your goodie. The PR You make a response, you get it, but then for the next one you have to work harder, and then it gets harder and harder and harder. And so that it looks something like this. There's different schedules or equations that are actually used. They become these exponential functions where you have to be working, just to get your next hit of drug or your next squirt of sucrose. And so it's thought to be a really good measure of motivation or effort beyond just what the simple fixed ratio schedule will tell us. What we've seen so far, we've been doing this work for a couple of years now, is that, at least in our laboratory, the female rats really like the sugar. The males like it too. The females like it a lot more. And these data, by the way, up here, are not, we're not incorporating body weights on here. The females are, so the males are about 1.5 times larger than the females. And so the females are much smaller than the males. Yet the females are consuming more. These are the deliveries of the sugar. Way more than the males. So if we actually corrected for body weight, these differences would even be bigger. We've done that, of course, but for these graphs, these are not with that change incorporated. So it shows up on this FR, it shows up on the PR. We've been doing a lot with different concentrations of sugar, seeing like if there's a concentration dependency to it on the FR across these different concentrations coming together at really high concentrations. The PR, not as sensitive to changes in concentration, but the sexting, these are all females, these are all males down here. We've also looked at the craving measure, the seeking behavior between males and females. These are the females. Up here, this is the control condition. We did a little bit of behavioral pharmacology where we gave them a drug that blocks a particular dopamine receptor, the D1 receptor, which reduces their seeking behavior. Does that to about the same extent for males and females. But you can see overall females much higher responding. And I can go into the stats later, but interestingly enough, it's not simply explained by the fact that they responded more during training. There's also a little bit more on top of that when they're confronted with the sugar cues. So almost to the last slides here. So back to the orbital frontal cortex. So we know males and females are different, at least in our hands in the laboratory. We've seen the orbital frontal cortex showing up in terms of activity of FOS and this dark protein and what it's doing related to enrichment, incubation. This is just going to be looking at it with enrichment. So can we do something to the orbital frontal cortex to negate the effects of enrichment? Maybe that'll tell us about what the orbital frontal cortex is doing that actually induces the anti-craving effects. So this is a functional study. So we're actually doing something directly to the brain. The technique is site-directed drug injection. So there's a lot of surgical stuff we have to do to be able to put a drug directly into these brain areas. We did some work with a region called the nucleus accumbens. A lot of people are probably familiar with that area. But also the orbital frontal cortex, you can't really see the blue dot so well, but that's where the injections went. We ended up micro-injecting. We did some stuff with the dopamine agonist, but the more interesting effect we saw was with a glutamate agonist. So we're stimulating, kind of activating the neurons with this drug called S-AMPA. And this is very, very, very piloty preliminary data. We'll come back and do some more on this, and I'm just highlighting over here in the orbital frontal cortex. So if we do vehicle, which is like the saline injection, if they've had enrichment, they don't respond very much for the sugar cues. We expect to see that. But if we give them enrichment and then we drop in the agonist on their glutamate receptors, on the AMPA receptors, then they responded like crazy. And we don't know exactly why. Was it making it so all of a sudden the sugar cues were now worth more because the orbital frontal cortex was like, yay, back on track? Or was it doing something else? Is this some weird side effect? That's what we want to explore with the grant. So this is the last slide here, is just specific aims. Taking that all together, sex differences like throughout. We know we have some. To explore sex differences, we're following kind of the typical kinds of things you would do. So with the females, we're doing studies where we track the estrous cycle, which is like the menstrual cycle in humans, but much shorter. It's like 5 days long. So we can then correlate the kind of hormonal states with their behavior in terms of their motivation to consume sugar. We also might be doing some studies where we gonadectomize, which removes like the ovaries, the testes. I know it's throw some people out there. To remove the influence of hormones, and you can actually put them back in a controlled manner. We're going to look at the persistence in a little bit more detail. We've actually been doing a lot of work on this already. How long do these EE effects last? Is it just a transient thing for one day, or does it last for several days? Look at sex differences, include abstinence, incubation, and then do more with the OFC. So do the stimulation, males and females, and get into some more of the detail there. Lots of people, as Dr. Grimm was saying, have been involved in these studies. I actually included all the way back because the slides I showed were data from postdoc. But any slide that had data, I hope I got everything right. People that were involved in that, on this laundry list here, I got some of the credentials of some of these people up to date. So I'm feeling old when I look at so I need some more MDs to come back and take care of me. I got one in the hopper right now. So, but yeah, that's it. That's my really fast talking. I want to respect the time, so it's almost 5. So I can take a couple questions, but then I can hang out if people have other questions. Anybody have like a pressing, interesting question? Yes.
So when I was in grad school, I spent about 3 years in federal prison, and on my clinical internship. And one thing that really struck me is how incredibly boring and how incredibly unenriching our penal system is. So we had a lot of folks in there who were in there for drug-related offenses, who were getting treatment while they were in prison, but just finding things to be able to pass the time while you're in there was painful for folks.
I could, horrible. Yeah.
So it makes me think about what are we doing to folks if we're having them abstain from drugs or alcohols in a period where they have an incredibly unenriched environment, and then we're sending them out into the world afterwards, kind of based on this research, depending on the length of time, possibly less prepared and worse off than they would be otherwise. It also made me think about how much of this has to do with enrichment and how much of it has to do with boredom instead, the lack of enrichment.
Right, the lack of it. And it's interesting you mentioned that before we ever started doing work with enrichment, so back when we were trying to come up with a name for the incubation effect, one of my bosses, Dr. Wise, was like, this is like for people in prison. And then they come out, so he wanted to call it like the incarceration effect or something like that. That sounds kind of boring. But I think you're totally right about that for people in that particular situation. There's a lot of other things too. I think there's like conditioning kinds of things that are different from the drug-taking environment when they come back and they haven't extinguished and things too. But just the fact that they might be in a situation where there's not a lot of activities or different kinds of things that might be stimulating, enriching, again, that's kind of trying to define what that means for a person. Could be very problematic. I agree. Yeah.
I might have missed it, but with the females, if you don't see the difference in extinction rate versus the males?
So there's other data slides. They are more resistant. Yeah. They just really like the lever and sugar and all that. Yeah.
How long did it usually take for the extinction, like how before the extinction of thing would happen?
So not having to do with incubation, but just like if you have a rat, and how long does it take to extinguish their behavior? We don't look at that specifically. We usually have like daily tests, that kind of a thing, and then over time. So with daily tests, it might take like a week to get down to a very low level of responding. I think if you put him in the box for many hours, just straight like that, probably over 8 to 12 hours, I'm kind of guessing based on some other data that we have, something like that. It starts to get very confounded because then they're the time of day and like their circadian rhythms and all this kind of stuff too. So when you prolong it like that. Cool. I'll take questions. There are 2 more cookies left. There's probably some coffee in there too. Thanks for coming and enjoy the